circular RNA as such as a therapeutic moiety is performing better than conventional mRNA because it is more stable. We dramatically increase the level of expression of a therapeutic protein, so it can allow treating patients which are currently have no choice. They have no treatment options.
Our coverage of Meeting on the Med in Rome is kindly sponsored by Cellares. I'm Federico Citteric, reporting for Onyx live here at the Meeting on the Med in Rome. Today I am with Victor Levitsky, CSO at Circio. Victor, thank you very much for joining us.
Nice being with you here. I've been trained as a medical doctor many years ago, and then had a long academic career at the Karolinska Institute in Stockholm, then at the Johns Hopkins University in the States as a faculty of the oncology department. And then I switched to the industry, was working for several big pharma companies like Roche, Servier in France, and then was developing portfolios of several biotech companies in Switzerland, in other European companies. And now already for like 6 years, I work in this role of Chief Scientific Officer of Circio.
And what is the main aim of Circio, what you guys do there?
Yeah, Circio historically started as a pretty typical immuno-oncology company. We were trying to treat cancer by stimulating immune system. But when we decided to diversify the technology portfolio of the company, and we brought in this technology, which is we refer as circVec technology. Essentially, what this technology does is to teach cells how to make circular RNA instead of conventional, more common mRNA molecules.
Why do you think circular RNA is gaining that much attention right now?
Yeah, circular RNA as such as a therapeutic moiety is performing better than conventional mRNA because it is more stable. And then a therapeutic product can be expressed for, let's say, 1 week or a little bit longer, in contrast to mRNA, which only gives you 1 day. But what is interesting that Circio is very differentiated from other circular RNA companies, because we are not making circular RNA as a product. We are making DNA vectors, expression systems, which teach our cells how to make circular RNA instead of mRNA. And this results in quite dramatic biological effects.
And what do you think needs to be proven so that circular RNA can be treated as a pragmatic option as well?
Yeah, we need more data in the clinic. And many products which are based on circular RNA, either directly, as some of our non-competitors are doing, or indirectly as we do, we need to accumulate more data, but the promise is very significant. This can be clearly seen from recent acquisitions of multiple biotech companies by big pharma companies for a significant amount of money.
Is this something you're discussing here at MoMed, perhaps?
Yeah, we of course would be interested in making licensing deals with big pharma companies, giving them maybe an aspect of our technology. And we have ongoing big pharma collaborations and several discussions with big pharma and biotech companies. But eventually, of course, we would also like to develop our internal portfolio of products for clinical application in the future.
And how do you think about delivery challenges? Is circular RNA facilitating things or making it more complex to actually physically get the thing into the patient?
Yeah, so what we are hoping to achieve is actually address some of the challenges which existing delivery instruments are facing. For example, the best case from our point of view is AAV, adeno-associated viruses, because at the moment it's the main instrument for gene delivery in the clinic. But use of adeno-associated viruses is associated with high toxicity and low efficacy. So what we achieve with our circular RNA endogenous production technology is that we dramatically increase the level of expression of a therapeutic protein from an adeno-associated virus. And this may allow to significantly decrease the dose, also pushing down toxicity, keeping the same or even better efficacy of therapeutic protein expression.
And finally, Victor, if this technology works as intended, what could it enable that isn't possible today?
Yeah, so it can allow treating patients which are currently have no choice. They have no treatment options. We will be able to deliver much safer therapeutics to them and probably expand this delivery, this therapeutic approach to diseases which currently are not treatable. So that's the dream. Or make the current treatment much more effective. That's as simple as that.
Well, that's also very important. Victor, thank you very much for joining us. Thank you again.
It was my pleasure. Thank you.